Thank you everyone for being here on Zoom. I'm really excited to introduce Professor Jasmine Nirody, Professor of Organismal Biology and Anatomy at UChicago and a member of the NITMB faculty. And we're really excited to hear what you say today.
All right, thanks, Alistair. Oops. All right, hello everybody. Thank you so much for having me here. I'm really excited to be a part of the series and that we get to start off this morning talking a little bit about adaptive biomechanics. So in our lab, we study how organisms move through natural environments, which are often dynamic in time and heterogeneous in space, and how their locomotive strategies have evolved to deal with this heterogeneity and variability. And to this end, we end up working with a lot of really interesting and agile organisms, so two of which I'm going to talk today, flagellated bacteria and Panarthropods like this tardigrade that I'm showing here. And I'll have more pictures and videos of them later, so don't worry. And it's probably pretty obvious that movement is vital to an organism's fitness. It dictates if an animal can escape from predators, how well it can hunt for food, how well it can find mates to reproduce, pretty much everything. And this is really salient across a broad range of timescales, right? So we're very used to this idea that organisms have adapted to the environment that they've evolved in. And this is partially why we are so concerned with the effects of climate change, of urbanization on biodiversity. But what's true is that locomotor systems also have to be flexible and mechanical performance has to be plastic within the lifespan of an organism. So for instance, animals that live in the tropics have to adapt their kinematic strategies when monsoons turn their environment from terrestrial to semi-aquatic. And bacterial species that spend part of their life cycles within other organisms. So for instance, a bunch of pathogens and this creature here, Vibrio fischeri, which is responsible for bioluminescence living in the light organ of the bobtail squid. And on even more kind of immediate timescales, bacteria and animals like tardigrades have to navigate these complex terrains like soil, or this guy here lives on moss, and they have to adjust their gaits and their movement strategies in real time as they encounter these variabilities and obstacles in their paths. And so our research focuses on questions that relate biological form and mechanical function across this wide range of timescales. And in the lab, we're currently focused on answering these questions using two broadly defined organismal systems, so flagellated bacteria and panarthropods. And this is a diverse group of animals that includes arthropods, tardigrades, and something called the velvet worm, which is as adorable as a tardigrade, I would say. And you can see a little snapshot of it there, and I'll zoom in later. But the reason that I've kind of arranged the projects in this way in our lab are to point out that there's a really strong mechanistic and technical difference as you go up this length scale. So for instance, when we're thinking about working at the molecular cellular level of biological organization, when we think about adaptive dynamics, we might refer to protein subunit remodeling or changes in gene expression. Whereas when we talk about these kind of things for multicellular animals with nervous systems, then we're instead talking about flexible behavior and sensory feedback mechanisms. And similarly, over here at the evolutionary timescale, we're looking at protein structure and molecular evolution versus morphological changes in body structure. And while the mechanistic differences across this chart are pretty massive, the reason that I want to talk about all of these together is that there's a unifying framework that we work with that joins all these together. Yes. Yeah, of course. Totally. Mm-hmm. Yeah. Mm-hmm. Sometimes you do. So for tardigrades, the picture that I had in the beginning, they walk under a thin layer of water. Yeah, so they get very easily dehydrated, so they're always walking on a surface but on underwater. So they kind of operate at incredibly low Reynolds numbers to that end. So basically all the questions that we ask and all the questions that were kind of across that chart arise from trying to comprehensively understand locomotive performance in real natural environments. And we think about these in different systems. And what this means is exploring both kind of how things work. So what are the underlying mechanisms? What are the structure function relationships in the underlying locomotive machinery? And also asking why. So what environments favor particular locomotive structures, particular locomotive strategies, and how have evolutionary constraints shaped the observed diversity of motility dynamics that we observe in a particular group? And then to properly pose these questions, we have to first start here in the center by trying to gain an understanding of the kinds of challenges an organism might face in the environments that they've evolved in, and characterizing how their motility dynamics actually look and how they change in response to these challenges. So I'm going to tell two stories of that today, and I'm going to start with flagellated bacteria. When most people think about flagellated bacteria, and when they think about flagellated swimming, kind of your mind might jump to this run and tumble system, right? And it's a clever way to be able to sense and navigate environments at the micro scale. So the way that this works is a bacterium will run for a short period of time, that is swim in a straight line. And to change direction, it'll initiate a tumble. And when it initiates a tumble, its flagellar bundle flies apart, and it kind of spins around for a short period of time and picks a new direction at random. So with the turn angles are uniformly distributed. And then it initiates another run, and so on and so forth. And the way that it can follow a chemical gradient is if it realizes it likes the direction that it's going in. I'm going towards food. I'm going away from some kind of toxin. Then I'll extend the length of my run. I'll tumble less frequently. If I dislike it, I'll tumble more frequently until I follow a gradient. So this way kind of follows a random walk with a drift. And a characteristic feature of this model of locomotion is that the mean square displacement curves show ballistic behavior at short time lags corresponding to the run duration, and giving way to long-term diffusive behavior. But then it's important for us to realize that, okay, we're talking about movement in liquid media. And with no confinement, no obstacles, they're just freely swimming as much as they want. And this is really different from most of the environments that bacteria are experiencing in their actual lives, right? They have to interact with our gut, they have to interact with soil, they have to interact with lots of different things. And recent work, for the first time out of the lab now at Caltech, looked at how bacteria move through some of these more confined environments. And they looked at very tightly confined porous media, like densely packed soils, for instance. And what they found was that motility looks quite different from the classical run and tumble. So the authors termed this motility to be called hop and trap. And you can see kind of pretty descriptive, because bacteria remain trapped for time, between which they can kind of hop between pore spaces when they find an escape. Yeah. Same species, both E. coli. Both E. coli. And for a long time, before these experiments, what was generally assumed was that when you put bacteria in crowded environments with obstacles, what would happen is that their run lengths would naturally get shortened, because they would keep hitting obstacles before they complete a run and then have to initiate a tumble. And so what this would result in is a shorter ballistic region, but dynamics that kind of overall mirror what you would expect from the classical run and tumble model. But in fact, that's not what we get when we look at hop and trap. Instead, we do see the shortened or close to non-existent ballistic region, but another surprising feature of hop and trap motility is this appearance of the subdiffusive regime at intermediate scales. And so this is a pretty radical restructuring of what we would expect from classical run and tumble dynamics. But these are also two radically different environments, right? They're totally unconfined environments versus really, really tightly confined porous media. And so we have to ask, and we ask, what happens in the middle? Are these two actually different locomotive modes? So that is, are bacteria behaving differently in these two environments? Are they doing something different at the individual scale? Or are these observe differences kind of purely driven by the environmental structure. So in our lab, Haibei Zhang, a graduate student, together with a collaborator, Miles Wetherington from Georgia Tech, spearheaded these questions experimentally by using a microfluidic setup that Miles constructed that consists of an array of PDMS pillars. And we can vary both the spacing of these pillars as a kind of a measure of confinement, as well as vary the variance of the spacing from how far they are from the grid as a measure of disorder. And you can see here an example of some fluorescently labeled E. coli moving through one of these mazes, which we can overlay with tracks. And what these experiments do is allow us to characterize what tracks look like in each of these environments. You can see here some comparisons with unconfined media and some of the structured environments. Yes. Yes, it's effectively 2D. It's quasi 2D, yeah. We eventually do want to expand this to 3D, but the difficulty is kind of tightly controlling the spacings and tightly controlling the variance. So is incredibly difficult to engineer in the 3D porous media. So the porous media, the previously is very tight and very variable, but the variance is huge. And so it's understanding kind of how, if we tune these parameters, you can get a rigorous characterization is not that trivial. Yeah. 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 Right. So what we found is that bacterial trajectories showing both run and tumble and hop and trap behavior. So you can see here the black one corresponding to this and the green highlighted trajectory corresponding to this coexist in intermediate environments. So the proportion of the trajectories that hop and trap behavior increase with both increasing confinement and increasing disorder. So basically, bacterial motility patterns seem to lie along a continuum, suggesting that this hop and trap behavior isn't fundamentally different from run and tumble at the individual cell rule level. So the individual agents aren't really behaving in a different way, but the environment is forcing the shift. And so using some simulations of a modified run and tumble model, we aim to kind of get at what are the parameters that are allowing us to tune and move along this continuum. And we first started with the obvious choice, which I already kind of talked about, tumble frequency, right? So like I mentioned, it's long been assumed in bacteria that when you have crowded environments, you're going to get shorter runs because you will bump into obstacles and you'll initiate more tumbles. And what this results in is, as you can see here, a shorter run length as shown in these run duration distributions. And accordingly, you get a shorter region of observed ballistic dynamics. But just varying tumble frequency doesn't result in the observed subdiffusive dynamics that we see at the intermediate time lags. And so there has to be something else going on here. And so we had a little bit of a think. And we realized that, okay, if a hop corresponds to a truncated run, then in this analog, a trap maybe can be thought of as a very long tumble. And what we hypothesize is that in confined regions, bacteria often can't initiate new runs successfully. And this results in many failed tumbles kind of being observed together experimentally as one super long expanded tumble or trap. And so adding this parameter of tumble failure into our run and tumble simulation does indeed then get this characteristic behavior that we observe from hop and trap. And albeit at very high probability, which is to be expected because this is a very extremal behavior. And actually the distribution that we see at the high is corresponding to the experimental distribution that we see in the porous media. But then we think about, so what's the mechanism here? How is this happening? We hypothesize that tumble failure results in these lengthened tumble periods, right? But how? Within the context of our experiments, tumble failure we found can occur in two ways. So first, highly confined environments can suppress the flagellar bundle from coming apart, which I mentioned in the very start is what's responsible for initiating a tumble. And so this prevents proper reorientation of the bacterium. And you can see that here, where the flagellar bundle is just kind of held in place. And a reorientation is initiated only when the bacteria able to fly its bundle apart. And then second, on the other axis, in highly disordered environments, obstacles can end up forming this kind of sort of dead end where you don't have a clear path forward. And so even if your flagellar bundle comes apart and you initiate a tumble, your reorientation just keeps you in place because you can't actually escape. And you can see that here, where you have a few bundles flying apart, but the bacteria is just kind of stuck in place because it's not reorienting properly. And finally, you can get out and you will eventually escape. There we go. You feel really bad for them when you watch these videos. Mhm. Yep. Yep.
All 60 degrees. It felt like it was sort of like biased towards going left as opposed to right.
I think that is large. So in these, because these are in these are videos from the 3D porous media. And so you get confined in because the pores are preventing you from going in particular direction. So in unconfined media, it's uniformly distributed.
Do coli have a mechanosensory receptor?
Their flagella are a mechanosensory receptor, which I will talk about in 2 minutes. So we now have spent a bunch of time talking about, and this is perfect transition, all the different things that the bacteria has to sense and respond to in its mechanical environment, which means that the underlying motility machinery should be equipped to be able to deal with this kind of these kind of environments as well. And so this machinery is a rotary molecular motor that sits at the base of each flagellar filament, and it's called the bacterial flagellar motor. And I'm not gonna go through every single protein in this complex, I promise. But just know that motors consist of 3 general parts: a spring-like hook that connects the body of the motor to the flagellar filament, a rotor, which is named as such because it spins with respect to the cell body, and a stator. And this is called that because it's stationary with respect to the cell body. And stators consist of these MOT units, which are connected via spring connection to the peptidoglycan layer in gram-negative bacteria. And each of these units have an ion channel that's associated with them, which is what allows the motor to couple ion flux to rotation at this rotor-stator interface. So basically what happens is with the passage of each ion, one of these MOT units will interact with the C-ring and push along one of these protein spokes, these flyg protein spokes, and make a single step around. And this, when I first started thinking about this motor, an analogy kind of helped me wrap my mind around its function. And the structure lends itself really nicely to that of a playground merry-go-round like this. So the rotor is ring-shaped. It has these protein spokes around. It's the merry-go-round itself. And we are the stators that are pushing it along to entertain our friends. And like the merry-go-round, the motor can go at a wide range of speeds. Now, this is data that's collected on E. coli from Howard Berg's lab at Harvard. And from the earlier part of this talk, you're aware that bacteria live really complicated, really exciting lives. And so this whole range of speeds and torques is relevant to a bacteria living its life. And so, for instance, when a bacteria is freely swimming in an unconfined environment, it lives here in the low torque, high speed region because the flagellar filaments are thin. They don't impart that much torque onto the motor. But when bacteria are interacting with surfaces, when they're interacting with other bacteria in a swarm, when they're trapped in a porous media, they end up over there quite quickly in the high torque region. And an important thing additionally is that gut bacteria like E. coli are not only dealing with both of these environments, but they often have to transition between them within their life history, and quite quickly sometimes. And what that means is that their flagellar apparatus has to be able to be equipped to make these transitions and to sense when they are making these transitions. So going back to the playground, how would we deal with these kind of switches? So let's say just our best friend wants a push on the merry-go-round. Okay, this is not too challenging, right? We can make the merry-go-round go fast. It doesn't make us too tired. Everyone involved has a lot of fun. But after a while, some of the other kids are going to realize that this is a lot of fun and they're going to want to come and join in. And now the required torque for spinning gets higher and higher and higher, and things are getting a lot tougher. So eventually, maybe you want to recruit some friends to help shoulder some of this effort. And it turns out that that's exactly what the flagellar motor does. So it was observed in E. coli that motors that are operating at high load have far more stator units that are engaged with the motor than those operating at low load. But we don't know much about the dynamics of these transitions. So how and when do you decide to call your friends over? How do they decide when to come over and help and how long to stay? So we started by constructing a mathematical model of stator engagement and disengagement to the rotor, characterized by an on-rate arrival rate K on and a off-rate disengagement rate K off. And what we asked was, is the stator arrival rate K on, leaving rate, or both, dependent on what the bacterium is experiencing, what the external load on the motor is? And to test this rigorously, we performed some experiments in the lab. And one way to get at motor dynamics and make a curve like this of torque-speed relationship is to shear off the flagellar filament, stick a bead onto the flagellar stub, and from here you can get out the speed of the bead using high-speed video or by using a feedback optical trap. And because these beads are really small, so the largest size bead we use here is about like 1 micron in diameter. And it's really easy to calculate out the drag on it. It's equivalent to the torque that the motor has to apply to get the bead of radius r to rotate at some speed omega. And so this gives us a picture of what the motor is doing at a particular load on this curve, denoted by this red bar. And the position of that is determined by what size bead we're using. Yeah, totally. Yeah. Yes. On one. So we tether the bacterium down to a slide by using polyol-lysine. So we just stick the bacteria down by its cell body. And so all its flagella are arranged randomly around the cell body. And so we have and they're all sheared off by the time we've sucked that bacteria down. And so you have a bunch of like stubs that are sticking out all over. And you flow beads on top. And beads will stick down. And usually you only get 1 bead per cell. So you will get, because the flagellar has to be, or the stub has to be oriented in a way that the bead will stick on it properly. Mm-hmm.
Saying, oh, in these higher load regimes, you're going to have to have more of these, what were they?
Stator units. Stator units, yeah. Yeah.
Because they don't and the fact that, you know, all the different flagella might be in sync or out of sync.
Totally. So the flagella being in sync or out of sync are controlled by an underlying chemical system. So that they're very tightly coupled with each other. Yeah. So there is like some stochasticity, but they're very, very tightly coupled. Yeah. So there's an underlying biochemistry system that's like keeping them very much in sync. Yeah. Yes. You have these like, you have this long tube and you have 2 syringes and you just go back and forth like 50 times. And eventually we also have done it genetically, which is much nicer. Because if you do this and you miss once, then it shoots out and goes right in your eye. Just as a warning if you ever want to try it. But yeah, so basically using a bead of a particular size gives us a point on, so each flagellar filament, so a bacterium has many flagellar filaments, each filament has a motor that's at the base of it. So we're looking at kind of a single flagella at each of these points. So what this gives us is a picture of what the motor is doing at a particular curve. And now you can move along this curve by, using a smaller bead, getting closer to the high speed region, using a bigger bead, getting us closer to stalling at high loads. But still, each of these points is giving us a static picture, right? It's giving us what's going on at a singular point on this curve and not much information about the dynamic nature of what's going on as bacteria are going back and forth across. However, okay, so if instead of using, the plain old beads that we were using, we used super paramagnetic beads, what this might do is allow us then to manipulate and perturb what the bacteria are feeling while the bead is still attached by using an external magnetic field. And so for this, we use 2 mounted permanent magnets that exert, a particular torque on the bead at a particular height. And then we can lower it so that the external magnetic torque exactly equals the torque that the motor is applying, so we can stall the motor for a particular amount of time. And before the application of this external forcing, you can see that there's a population average of motor that's spinning along with a fixed number of stators. And then we lower the magnets into position, keep them there for 5 minutes. And after this period, we raise the magnets again to see what the motor has been doing. And you can see that during this time where we applied this lot of external torque, the number of stators jumped up. And that indicates that the motor underwent some remodeling in real time. So once the torque is removed, the motor again undergoes some subunit exchange and relaxes back down to the original stator number for this bead size. And from this relaxation process, we can get out the characteristic time, the steady state stator number, and then we can plug it back into the kinetic model that I described earlier to get out an idea of the engagement and disengagement rates, K on and K off. Yes?
Curve is like you're averaging over
Averaging over multiple bacteria, basically. I'll show later when we can kind of remove some of that variability by looking at individual bacteria. But at this stage, because we're doing these kind of permanent magnets, we have like a whole field of view at a time. And so we can repeat this across different bead sizes, which then allows us to get an idea of how these rates look across this curve, right? So if the magnets pull us over there, close to the maximum torque of the motor at 0 speed. Then once the beads are released, the relaxation processes take us to these various points along this curve determined by the bead size. And the issue here is that we couldn't quite access the low torque region using this experiment based on just the smallest commercial bead size that was available to us. And this was an obstacle because, like I mentioned, performance across this whole curve is really important for bacteria living their life. And we wanted to kind of understand what's going on across this entire curve, particularly this, very dynamic region here. So again, we had to have a bit of a think. And what we realized is that using permanent magnets, we're able to go in this direction to stall the motor and making us completely dependent on bead size. But instead, if we use a rotating magnetic field, we can spin at any speed the bead and the motor to follow along on a single cell. And so what this does is allows us to go kind of back and forth along this curve using one bacteria and removing some of that variability, that intercell variability. And luckily, this includes the low torque region. So on the right here, now instead of showing a population average, I'm showing a single cell trace. And you can see it's moving along happily about 45 Hertz without any interference from us. So this is before the magnet has been turned on. Speed jumps up to 200 Hertz because that's the speed that this external field is going at in this particular trial. And when we turn it off to see how the bacterium is doing, you can see there's already some remodeling going on. We do this again and again and until we get a clear time course for how stator units are behaving at this particular load and particular speed. And you repeat this across many different loads, across many different bacteria for each load to get some statistics out for the parameters that allowed us to calculate on and off rates across this entire curve. Yes? Oh, it's spinning because well, it's sad. So this is another thing where you feel really bad for the bacteria because you've tethered the bacterium down. You've put it in a it thinks it's swimming, but it's not. It's very sad. I've developed like a lot of weird empathy for bacteria. Yeah, basically. I mean, it just wants to go, right? So it thinks it's swimming. Yeah, so we can get out the kind of parameters for how the motor is remodeling across this whole curve. And putting all this together, what we found is that the on rate, which I'm showing here in blue, actually didn't vary across load conditions, but the off rate, which I'm showing here in red, decreased as it increased. So essentially, stator units are coming by to check on their friends regularly, despite how tough the job actually is of pushing the motor. But when they come by and they see that there's a little bit of a struggle, they stay for a bit longer. And this kind of bond, which grows stronger with larger external force, is called a catch bond. And it's a pretty widespread molecular strategy across different systems, both prokaryotic and eukaryotic systems. So in leukocyte adhesion, bacterial adhesion, and then in actomyosin systems. And also, it's familiar to us at the macroscopic scale. If you remember this toys from our youth when we would have these finger traps. You put them in, and you try to pull your finger out, and it only tightens, and the only way to actually escape is to relax and let the bond go slack. And so this strategy is one that took me a while to learn when I was 5, and took the flagellar stator longer over evolutionary time to learn as well. Now, we talked about all of these experiments that I showed you in understanding kind of how the E. coli machinery works in these different environments. But we also want to think kind of more broadly across bacterial species, because there are a wide range of flagellar structures, flagellar organizations that exist across a number of pathogens and environmentally very significant species. And what's really exciting about this diversity is at its core, the flagellar motor structure actually is quite well conserved across all bacterial species that have a flagella. So you can see here in solid colors the bits that are evolutionarily conserved across all these species. And nicely for us, this includes the stator and the rotor and the stator-rotor interface. So we can ask a lot of questions here about how exactly this mechanosensitivity might have evolved across bacterial species, in particular in bacterial species that don't necessarily have to live in really complex environments like our guts. Marine bacteria, do they maintain this level of mechanosensitivity? Have they changed this method in any way? Because what are the trade-offs? Is it cost effective? Is it efficient in any? Does it create any sort of energy cost on the bacterium that might be good to lose in particular lineages? So in the lab, we're looking across a bunch of different bacteria that have evolved in a bunch of different environments to see how mechanosensitivity looks across these motors. And one of these bacteria is Vibrio fischeri, which is the one that I showed at the very start, that lives in part of its life within this bobtail squid, and moves within this kind of very tightly packed light organ. And so the interesting thing about this bacteria in particular is that it spends its evenings within this light organ and its daytime within the marine environment. So every day it kind of has to switch between these two radically different environments. And so we ask, what does this flagellar machinery, what does this bacterium do to be able to be successful in both of these types of environments? And like I said, in comparison to bacteria that perhaps live in kind of a singular type environment. All right, so before I jump up to the other set of systems, maybe I can just pause for a second and ask if anybody has any further questions on the bacteria stuff, or you can save them until the end. Yes.
So signal transduction.
Yep. Yep.
That when it does that, does it cause the stators to fall off?
No. So the stator stays stuck on. So the chemosensory machinery actually only affects the rotor. So you have the spokes that point in a particular. What the sensory machinery does is it switches the position of the angle of the spoke, so that even as the stators are moving in the same way, the motor reverses direction and causes a change in the torsion in the flagellar filament, which gives you a kink and then makes the bundle fly apart. All right. So jumping up to Panarthropods, I'm gonna talk about tardigrades, and I've been completely taken with tardigrades for a long time now. So it's been really satisfying watching them get a lot of attention in recent years in the media. So even if tardigrades aren't your system of choice, maybe you've heard about them because they are remarkable in their ability to survive in a wide range of environmental extremes, right? So within tardigrada, there's that can survive in extreme temperature and extreme radiation, really almost anything. And even recently, I think not recently, I think this is like on used for crash landing on the moon. And I think we're gonna go back and check if they're alive in 100 years or something. So we'll see. TBD. But I think that their amazing survival skills are range on Earth. So like flagellated bacteria, tardigrades have thrived in just every environment imaginable, essentially. Marine environments, freshwater environments, terrestrial environments. And then they've managed this kind of remarkable ecological success with a pretty evolutionarily conserved body plan and locomotory apparatus. So they have 3 pairs of clawed walking legs and 1 pair of reduced backwardly attached hind legs that they use to as hold fast, essentially, if they want to get some food from their water environment. And tardigrades can really, yeah. So the species that we work with is between 100 and 200 microns in length. And the kind of largest species that are known are a little under 1 millimeter. And then there's slightly smaller species, but the ones that we worked with are on the smaller end. And so if we ask, like, what can tardigrades teach us about walking, and why is it valuable to look at them? So like I mentioned, they're very small. So they give us this idea of what scaling looks like in walking kinematics. What do the smallest walkers do? And in particular, what do the smallest walkers that walk through water do, which puts them at incredibly low Reynolds numbers? So I sometimes hesitate to say that they're the smallest walkers because there are these mites, which are incredibly, incredibly tiny as well. But mites run incredibly fast and do not walk underwater. So I am pretty confident in saying that tardigrades are the lowest Reynolds number walkers. And furthermore, they're soft-bodied. They don't have a rigid endo or exoskeleton. And so when we think about all the techniques that we have to understand walking kinematics, they all, like I'm showing here in Drosophila, right? So they all kind of come from this ability to track the position of discrete contact points, like the legs. So you can, get out phase differences between leg pairs. You can get out stepping patterns. You can get out stepping time, swing relationships. And all of these are possible because you have these discrete contact points. Now, in soft-bodied crawlers like earthworms or nematodes, you can't really get this because you have a continuous line that's attached to the earth, and you don't really have a clear idea of what gates mean in these organisms. And the reason that I kind of know this very acutely is I tried at one point to get out similar kind of gait data from snakes. And I'm showing here movement on photoelastic gelatin. So you put an organism on gelatin through cross polarizers, and you can get an idea of the deformation. And what that gives you is where the animal is pushing down and when. So it gives you some idea of what their gait pattern looks like. And this was incredibly hard for lots of reasons. One is that, every time you make the gelatin, it's not regularized, and it's very hard to kind of get any sort of quantitative analysis out of this. And the second is that snakes and other animals don't really like being on gelatin, and they will not behave the way that you want them to behave, and also they'll bite you. So it's not very nice. But tardigrades are a different animal because they have this soft body, and they give us this kind of porthole into understanding soft body locomotion using techniques that we can use on skeleton animals. And the way that we work here is a very similar setup like the one I showed for Drosophila. We have a camera, and we put them on agarose gel, and this is gel of about stiffness 55 kilopascals, and we can image them from both side and top views. And the data I'm showing here is from this species called Hypsibius exemplaris, which is a moss-dwelling species, so one that has to kind of move through environments that are constantly changing and quite rough. So once we had the setup up and running, and that allowed us to get out this kinematic information, the first question that we asked was, okay, are they actually doing anything regular? And the reason that this is a reasonable question to ask is because tardigrades have about 200 neurons. So it's a really, really simplistic system. And we wanted to understand if they were actually as clumsy and silly as everybody thinks they are, as their name even indicates, right? And what we found is that actually tardigrades consistently walk with a very standard stereotype pattern that's tetrapod-like. And it's called that because diagonal legs swing or lift off together in sets of 2, leaving 4 legs, a tetrapod on the ground. And then we measured the relative timing between contralateral leg pairs, so leg pairs that are directly across from each other on a singular segment, as well as ipsilateral leg pairs that are on the same side on adjacent segments. And what we found is that tardigrades preferred the same coordination with an ipsilateral phase of about 1/3 and antiphase contralateral coordination, so kind of alternating contralateral legs across all the walking speeds that we measured. And what was particularly exciting about this is that these patterns are strikingly similar to those that we observe in arthropods that are many scales of magnitude larger and also have completely different skeletal structures and completely different ecologies. And so this is another reason why we were really excited about exploring how they might be able to sense changes in their environments, because you see the same things that you see in these other creatures. So what we did was we started thinking about how they might be able to deal with changes in slipperiness or flow in their substrate. So we made the gel slightly softer so that there would be more slip as they were walking forward. And this particularly we thought is quite salient for a soft-bodied organism, which doesn't have a skeleton to use to propel itself forward. So the way that we walk is when we put our leg down, I use my skeleton, I use this leg as a catapult to move my center of mass forward. But if I do not have a rigid skeleton, this is a noodle, and I can't pole vault with a noodle. And that's what tardigrades are doing essentially. So they're instead of pole vaulting, instead of walking, they're using their claws as grappling hooks. So they kind of go into the ground. They use the ground as their skeleton to pull themselves up. So they're essentially climbing on flat ground. If you're doing that, and then I make the ground soft and I give it a lot of slip, what do? How do you adapt your motility machinery to that? And what we found was, in fact, they do change their pattern. So you remember that their contralateral pairs on the stiffer substrate would alternate like how I'm alternating my legs. And you can see now that instead they're galloping. They're kind of going both legs at once. And like I said, one thing that was very exciting about this is that this is remarkably similar to strategies that we see in beetles when they go onto sand, as well as in millipedes when they go from walking to. So whenever something is dealing with granular or slippery substrates, we see this shift across the arthropods. Yes. Yeah, so they are being used, but the motion is so variable with respect to the other ones that it's very difficult to say kind of how they correlate to any sort of locomotive metric. I spent a lot of time like looking at that data and seeing if there was any signal in that noise, and it didn't seem to me that there was, but I would be happy if somebody wants to like play around with it and see if there's anything in there.
So you're saying there's like two modes of motion.
Yeah. Yeah, basically.
Switches.
It seems like this is true. Yeah, yeah. It's not, so that's what's really interesting. So that's what's really interesting about this and what's been characterized in beetles and in millipedes and everything. So is that the galloping mode is actually slower. And then that's partially because of the slip. But even when accounting for the shorter, kind of shorter step length, because you get a slip with every step length, even accounting for that, the movement is slower. Like each time, or the time for each step is longer. So the gallops are somehow like more careful because they realize that they're on the slippery terrain. Yep, it's totally different. Yeah. Because that's a transition with speed. So you put a horse on the same terrain, and you make it speed up in some way, it will naturally transition to a gallop because it hits a speed ceiling with a amble or a walk. And you have to enter a gallop because at a certain point you can't have all your legs on the ground and go faster. So you have to enter this kind of more aerial phase. For these guys, they don't care about their speed. They're more concerned with not falling. All right, so now we wanted to think about kind of, okay, we know that they're doing this. Where is this coming from? What can we say about the underlying neural structure? What can we say about the underlying mechanism or model that drives this kind of motion? And with all of this together, we can propose this really, really simple underlying control circuit, which consists of mutual inhibition between contralateral leg pairs and a back to front inhibition on each ipsilateral side. So a natural place for a circuit like this to exist is in the ventral nerve cord, or the VNC. And this consists of, for tardigrades, 3 segmented ganglia, 1 for each leg pair. And each of the ganglia is divided into 2 hemiganglion, which are kind of loosely connected. So essentially, each of these hemiganglia correspond to a leg circle here on the right. And within kind of an evolutionary context, which I've been hinting at a lot, is that there's this huge range of tardigrade species. We don't really know beyond kind of this gross structure how tightly their locomotive behaviors are constrained across this, and it's something that we're very interested in looking at. But also that the general topology of this VNC, the gross anatomy of the nervous system, is conserved not only across tardigrades but also with arthropods, which is kind of is exciting to us when we see these functional similarities across these species that we've seen. And one of these in particular, we were able to look at a large kinematic data set in Drosophila. And there was a study that mapped kinematic parameters onto this low dimensional manifold. Now I remain a bit like, this is difficult for me. This particular projection is difficult for me to interpret. I'm not very keen on like understanding this, like trying to interpret the structure of this. But what I do know is that if you throw in our data with the Drosophila, it's indistinguishable. So the tardigrade data falls very nicely onto this manifold. And then exciting is kind of developing more kind of mechanistic-driven models to understand the links between tardigrade walking, between these large kinematic data sets that we have in Drosophila and others, and partially in developing some of these data sets, in particular across different types of terrain. So in my lab, a postdoctoral scholar, Aaron Brandt, and an undergrad, Maria, are working on developing these kind of data sets in the drainfly. This is a long and papers of different roughnesses, as well as in jumping spiders. And you can see them walking here. They're reluctant, but they will walk. As well as on using that photoelastic gelatin that I spoke about earlier. So you can see, I don't know if you can see it very clearly on the screen, but you can kind of see the rays of light coming out of each of the legs as they push down. Because we're forcing it to not jump. We've put it inside a constrained environment. It wants to jump, which is why it really doesn't want to walk. But we also, so we're using this photoelastic technique for jumping. And in particular, one of the things that we're very interested in here is looking at the leg pair specialization. So a lot of these spiders do very elaborate sexual displays, which means that certain leg pairs are kind of veered away from having the musculature for good walking. And so we want to understand how does that, or good jumping, how does that affect their jumping ability? How does that affect where they're pushing down? How does that affect how they kind of readjust their kinematics to accommodate? All right, so that's all, and I want to say thank you to many of the fantastic collaborators that have helped with this work, in particular for the swimming work at the very beginning. Cody was involved with a lot of the and theoretical work, and I'm happy to take any questions. Yes. Yep, sir.
So this is fascinating. Interesting. I'm wondering, like, are there open mathematical questions that you don't know how to approach that might be kind of a good way to engage in NITMB?
Yes, absolutely. So a lot of, oh yes, so Rick asked, if there are any good like any kind of salient mathematical questions that this work can inspire to engage the NITMB community. Yes. So in particular, we've been talking a lot with Alistair about some of our how our data can be interpreted within the concept of these graph models. In particular, the walking data. So to understand connections between different leg joints, between different pairs, and to be able to kind of get that out immediately and to do gait identification in these different organisms, and to kind of map them onto each other, and to understand kind of universality. If you have a graph structure, and Alistair will tell you much more detail about this, but if you have a graph structure that looks at leg pairs across an organism and across different points, then you can look at 6-legged animals and 8-legged animals and say your contralateral leg pairs are showing this particular relationship. You're showing a relationship between these, and those methods to kind of get that sort of universality are just not there.
So this gangland picture, should I think there's like some kind of central?
Yep. So that's an idea. The idea is that each leg corresponds to a central pattern generator, but we don't know yet, and the mathematical modeling for that also is not yet strongly developed. Yep. We do. Yeah, totally. Absolutely. We do that all the time. So we physically, one thing that we can do is cut off a leg. So that's a very easy way to go about it. But using genetic knockouts, we can break any bits of the ganglion. We can break connections as well in the neural connections. Yeah. Yes. Right. Yeah. It's a weird quirk of evolution, right? Because we evolved bilateral symmetry like quite early. So like most things have even numbers of legs, even though I think like a tripod is the best thing to have technically in performance-wise. But that the system within like sea stars and other non-arthropods would be very, very different. I don't know what the underlying neural structure looks like there. And I don't know that it's well studied, but I'd be very curious to know what it does look like. Because the system that I'm showing in arthropods is very different from the system that you see in vertebrates, for instance. So even though we have a horse with 4 legs, a horse's nervous system and the connections within it are completely different than you'd see here.
Like they're primitive. I mean,
Yeah. When these guys came about, I mean, primitive is a difficult thing to say, right? Because everything has evolved for the same amount of time. But yeah. No, I know. But everything has kind of evolved for the same number of years. But they're certainly simpler. They're smaller, and so you have to have a much simpler system because you have a smaller number of computational units, right? You have smaller numbers of neurons, so you can't do as much with our brains or a horse. So that's particularly why they're so fascinating, because you can have this rich repertoire of behaviors using a very simple underlying control system. And towards kind of, even with tardigrades in particular, what the minimal systems are to be able to generate a set of behaviors that can complement and dynamic environments in particular. So in that sense, yeah, definitely I would say functionally probably more primitive. Yes.
Interval in human gaits where the interval is not completely uniform along. There is a certain variation.
Mm-hmm.
That variation we call the power law
Mm-hmm.
distribution. Do you have the data for your target?
Do you mean the variation in kind of stepping times? We do have the data, but we haven't looked at what the exponent would be of a power law there. But that's an interesting question. I don't know if how it would relate to the human one. I don't even know how it would relate to, for instance, Drosophila or other arthropods. It's a good question. Yeah.
you had these few slides talking about the ganglion structure and how certain inhibitions.
Yeah.
I was just wondering, like, how much of that is a model and how much of that is like confirmed? We know that
Oh.
that acted that way, and like
All of it is a model, is the answer. All of it is a model. We are working towards understanding the connections. So no. So even in insects, this is a model, because getting clear evidence that there's a central pattern generator is incredibly difficult. The way that you get at this experimentally in insects is you can cut off a leg, and then you can initiate fictive patterns and see if you get kind of oscillatory behavior in the leg on its own. And the conclusions are very shaky there. It happens sometimes, it doesn't happen sometimes, it happens in some species, it doesn't happen in others. And it's that have the same behavior. So it's not really clear what's going on.
like that.
No, because I mean, unfortunately, Drosophila doesn't have 200 neurons, right? So it has way more, and I think it's only very recently that we have any kind of connective map in any insect or any arthropod, and getting at kind of underlying models from that again is an interesting mathematical question, and I would relish anybody working on something like that. No, I would love for it to be done, but it's I think we're so recently into having a connective model for any sort of any insect or any arthropod that it's probably underway somewhere, and I would be very happy if it got underway here, but not yet. Yes.
There's a bunch of methods developed for human gait analysis.
Mm-hmm.
Analogous for studying.
So I would hope so. So eventually, I hope that that's one of the things that I'm hoping will kind of come out of being at NITMB, is that we develop better methods to do classification and understanding how to more quantitatively characterize these gaits, because we don't have a clear understanding of what constitutes a gait even. So there has a long history of treating arthropod gaits like you treat human gait, like you treat horse gaits, and they're quite different, because you see this continuous transition in arthropods that you don't see in let me go back to this. So regardless of what I think about the particular structure of this model, the point is that there is a continuum of gaits here. So there's no discontinuity like you would see for horses. So a horse walks and then it transitions to a gallop, and there is a clear switch in underlying control pattern there. Arthropods don't show that. When you're walking on flat terrain, unless you're giving something that forces a sensory feedback mechanism that switches you out, you have a continuum of gait patterns. And whether that actually means that these are individual gaits, or if you just see a single gait that changes continuously with speed and then is perturbed in different ways and then goes back to an underlying steady state is unknown, and is something that requires a pretty intense amount of mathematical modeling.